Okay, let's think about this. What's your absolute favorite song? Or maybe your favorite food?
Yeah, those things we love.
How much do your senses, drive those choices? And how much is that tied into just who you are, your identity?
It's fascinating, isn't it? We just sort of use our senses all day, every day. We don't really stop to think about the incredible complexity behind it all.
Mhm.
They are quite literally our connection, our pipeline to the world around us.
And that pipeline, exactly how it works, that's what we're diving into today. It's all about these amazing processes connecting the outside world to, well, your inside world, sensation and perception.
Right. And we've got this great set of source materials you've provided to help us navigate it. Think of this whole deep dive as like a shortcut to understanding that difference.
The difference.
The difference between just getting the raw data and actually making sense of it all.
Yeah, okay. So our mission here is to pull out the really key ideas from the material. Hopefully by the end, you'll have a much clearer picture of that split, just sensing things versus actually perceiving your reality.
Definitely. And we'll hit the big ones, seeing, hearing, touch, taste, smell, but we'll also get into those other senses, the ones that keep you upright and tell you where your arms and legs are.
Okay, let's unpack this then. The world is just completely packed with stimuli. Light, sound, textures, objects, events, you name it.
And a really fundamental point the sources make right away is that you don't experience any of that directly. Not really.
What do you mean?
Your senses pick up information about what's out there, and then your brain has to take that information and actively build your perception of it.
Okay, so step one is sensation. That's the initial bit.
Exactly. It's the process of receiving those stimulus energies, light waves, sound waves, heat, pressure, whatever it is, and then, crucially, transforming them
Transforming them into what?
into neural energy, signals your brain can actually work with.
Ah, okay. So specialized cells in our eyes, ears, skin, nose, tongue, they do this conversion.
Precisely. These receptor cells detect their specific type of energy and turn it into an electrochemical impulse, an action potential. That's the language the nervous system speaks.
And that action potential, that signal, travels along sensory nerves, carrying that raw data up towards the brain.
And that's where perception comes in. This isn't passive reception anymore.
Right.
This is the brain getting involved, actively organizing and interpreting all that incoming sensory information. This is how the raw signals start to mean something. A recognizable face, a familiar voice, the smell of coffee.
The sources had a good example. Sensing, like a silver object moving fast in the sky. That's sensation.
Yeah, just the raw input.
But perceiving it as a jet plane, that's perception. And they really stress these aren't two totally separate things happening one after the other. They're intertwined.
Constantly interacting. Like feeling a hug, you sense the pressure and warmth, sure, but you perceive it as comfort, connection. Sensation provides the data, perception gives it meaning.
Which leads to these two key ways of processing information the sources talk about, bottom up and top down.
Right. Bottom-up processing is really driven by the stimulus itself. It starts right at the sensory receptors. They register the info, send it up to the brain, and the brain then tries to piece it together.
So it's data-driven.
Yeah.
Like taking in raw ingredients and figuring out the recipe.
Exactly. A great example is hearing a song for the very first time. You're just processing the notes, the rhythm, the sounds as they come in. No prior framework for it yet.
Okay, then top-down processing is the opposite, starting from the brain.
Pretty much. It begins with your existing knowledge, your expectations, the context you're in. You use that higher level cognitive stuff to influence how you interpret the sensory data coming in from the bottom.
So using the music example again, if it's your favorite song.
Right. Your brain instantly recognizes it. It applies that favorite song framework you already have, based on memory and experience, to the sounds hitting your ear. You're hearing it through the lens of what you already know and expect.
And the key takeaway is that we're almost always doing both at the same time, right? They work together.
Absolutely. It's a constant interplay. Your eyes take in light patterns, that's bottom up, but your brain uses your knowledge that you're looking at, say your dog, to interpret those patterns quickly and efficiently. That's top-down influence. Makes the whole system work better.
So why? Why do we have these intricate systems? What's the evolutionary angle?
Survival, basically. Adaptation. The sources emphasize this evolutionary perspective. Organisms need to sense what's going on around them quickly and accurately to survive and reproduce.
Spotting danger, finding food, finding a mate.
Exactly. Is that shadow a predator? Is that berry safe to eat? And you see incredible adaptations in sensory systems across species. Predators often have eyes facing forward, gives them great depth perception for hunting.
While prey animals often have eyes on the sides.
Right, for a wider field of view. To spot danger sneaking up from almost any direction, the sources even mention this fish, the Anableps
The
microlepis.
four-eyed fish, yeah, I saw that.
Wild, isn't it? Two eyes looking up into the air, two looking down into the water, perfectly adapted to its niche right at the water surface.
But here's something really interesting the sources highlight. Subjectivity.
Ah, yes. Huge point.
Even if two people get the exact same physical input, the same light waves hitting their retinas, the same sound waves hitting their eardrums, their perception can be totally different.
Totally. It's why you can love a certain food or piece of music and your friend just shrugs. The sensation, the raw input might be identical, but their brain, with its unique history of experiences, preferences, expectations, it constructs a different meaning, a different perception.
It really underscores how personal our experience of the world is.
It does, and at the very start of all this are the sensory receptors. The material calls them the openings through which the brain experiences the world. I like that.
Yeah, me too. Figure 3.1 lays out the different types based on energy.
Right. Photoreception for light, mechanoreception for things like pressure, vibration, movement, so touch, hearing, balance, and chemoreception for smell and taste, detecting chemical molecules.
And figure 3.2 shows that basic flow. Energy stimulus hits a receptor cell.
Which converts it, that's transduction, into an electrical signal that triggers a sensory neuron.
Which fires an action potential, the signal heading to the brain.
And an interesting detail there is about intensity. The receptor's initial response might be graded stronger stimulus, bigger initial signal, but the action potential itself traveling down the neuron.
That's all or nothing. It either fires completely or not at all.
Right. So how does the brain know if a light is dim or blindingly bright if the signal strength is always the same?
It's the frequency of firing, isn't it?
Yeah.
More frequent action potentials signal a stronger stimulus.
Exactly. A brighter light makes the visual neurons fire more often. A louder sound makes the auditory neurons fire more often.
And how does the brain know if it's seeing something or hearing something if it's all just action potentials?
That comes down to specialization. The receptors themselves are highly selective photoreceptors, only respond strongly to light, auditory receptors to sound vibrations, and crucially, they send their signals along different dedicated neural pathways to distinct processing areas in the brain. The pathway tells the brain the type of information.
Okay, makes sense.
Oh.
But sometimes the sources say these pathways can get tangled, like with synesthesia.
Yeah, synesthesia is fascinating. It's relatively rare, but it's where stimulating one sense automatically triggers an experience in another sense.
Like seeing colors when you hear music, or tasting shapes.
Exactly. The sources mention someone who tasted tuna fish when hearing music. Neuroscientists are still figuring it out, but it might involve some unusual connections or cross-activation between different sensory areas in the brain. Maybe the wiring's just a bit different.
And then there's phantom limb pain. That seems related to brain wiring too.
Very much so. It's surprisingly common. Up to 95% of amputees report feeling sensations, often pain, in the limb that isn't there anymore.
How does that even work?
Well, the brain areas that used to receive input from and control that limb are still there. They can become active, maybe spontaneously, or maybe due to input from nearby areas, and the brain interprets that activity as coming from the missing limb. It's getting confusing signals.
And that's where things like mirror therapy come in.
Right. Using a mirror to create the visual illusion of the missing limb being intact can sometimes relieve the pain. It's thought to work by giving the brain updated, congruent visual feedback, helping to resolve the conflict between the phantom signals and the reality. It really shows how interconnected our senses are and the interplay between bottom-up signals and top-down interpretation.
And speaking of pathways, most of this sensory info, except smell, goes through the thalamus, right? Like a central sorting office.
Yeah, the thalamus acts as a major relay station. It receives sensory input and directs it to the appropriate areas of the cerebral cortex for higher level processing. Occipital lobes for vision, temporal for hearing, parietal for touch, pain, temperature.
And crucially, how we finally interpret those signals isn't just based on the raw input. Our learning, our goals, our expectations, even how alert we are, those top-down factors heavily shape the final perception.
Absolutely. It's an active construction.
Okay, so we need energy to sense things. But how much energy? That brings us to thresholds.
Right. The most basic is the absolute threshold. What's the definition there?
It's the minimum amount of stimulus energy someone can detect 50% of the time. Any less than that, and it's basically undetectable, or at least not reliably.
Think of the classic ticking clock example. You move away until you can't hear it, then edge closer. That point where you just start picking it up about half the time you listen for it, that's your absolute threshold for that sound in those conditions.
And the sources point out just how sensitive our senses can be under ideal conditions. Figure 3.4 has some amazing examples.
Yeah, seeing a candle flame from 30 miles away, detecting one drop of perfume in a 3-room apartment, our systems are capable of incredible sensitivity.
But the real world is rarely ideal, is it? There's always other stuff going on.
Exactly. Which brings in the concept of noise. And importantly, in this context, noise isn't just sound. It's any irrelevant competing background stimulation that makes it harder to detect the signal you're interested in.
Okay, this also touches on subliminal perception. Can things below that absolute threshold, things we're not consciously aware of, still affect us?
Ah, the controversial topic. The sources are clear that the famous James Vickery eat popcorn, drink Coke experiment was a hoax. He never actually did it effectively, or the results were faked.
Right, but that doesn't mean subliminal influence is impossible.
No, more careful lab studies have shown effects. The brain can register information presented too briefly for conscious awareness. And sometimes this unconscious registration can subtly influence thoughts, feelings, or behaviors. The study about flashing thirst-related words.
Yeah, where thirsty people shown those words subliminally drank more afterwards.
Exactly. It suggests that while we're not puppets controlled by hidden messages, information below the conscious radar can sometimes have a measurable, albeit usually small, impact. It links into priming effects.
Okay, so that's detecting something. What about detecting a change?
That's the difference threshold, or the just noticeable difference, the JND. It's the smallest difference between two stimuli that you can reliably detect, again, about 50% of the time.
Like telling two slightly different shades of paint apart, or noticing if someone added a tiny bit more sugar to your tea.
Precisely. And the key thing here is that the JND isn't a fixed amount. It depends on the intensity of the original stimulus.
How so?
It's easier to notice a small change in something weak than the same amount of change in something strong. Adding 1 pound to a 5-pound weight is noticeable. Adding 1 pound to a 50-pound weight, much harder to detect.
So the louder the music is already playing, the bigger the volume change has to be for you to notice it.
Exactly. And this relationship is captured by Weber's law. It states that for two stimuli to be perceived as different, they have to differ by a constant minimum percentage, not a constant absolute amount.
The candle example in the sources illustrated this well. Adding 1 candle to 20 is noticeable, maybe a 5% increase. But adding 1 candle to 120, that's less than a 1% increase, probably not noticeable. You might need to add, say, 6 candles to the 120, keeping that 5% ratio to see the difference.
Right. It's about proportional change.
Okay, so we can detect things, we can detect differences, but the world is just bombarding us. How do we manage that sensory overload?
That's where the active processes of perception really come into play, starting with attention.
We can't possibly pay attention to everything at once.
No way. So we use selective attention. We focus our awareness on specific aspects of our environment while filtering out, or at least turning down the volume on everything else.
The classic cocktail party effect, right?
Yeah.
Being able to tune into one conversation in a noisy room.
Exactly. And your attention isn't rigidly fixed, it's shiftable. You might be focused on that conversation, but if someone across the room shouts your name, your attention snaps over there. We're always monitoring the background to some extent.
And some things just grab our attention automatically, don't they?
Definitely. Novel stimuli, things that are really big or brightly colored or moving those, tend to capture our attention whether we intend them to or not. Survival advantage, probably.
But focusing really hard on one thing can make us miss other things.
Yes, that's inattentional blindness. When your attention is intensely focused on a particular task, you can fail to notice other unexpected things, even if they're really obvious and right in your field of view.
The gorilla study. Simons and Chabris, people counting basketball passes, completely miss a person in a gorilla suit walking through the middle of the scene.
It's a stunning demonstration. It shows that simply looking isn't the same as seeing. If your attention isn't directed towards something, you might not perceive it at all. And the sources note it's more likely when the main task is hard or the unexpected object is very different from what you're focused on.
Another big influence on perception is perceptual set. What's that about?
It's basically a predisposition, a readiness to perceive things in a certain way. It's based on your expectations, your prior experiences, your context. Like putting on psychological
That's
filters.
a top-down thing again.
Absolutely. The example with the playing card showing people specially made cards like a red ace of spades. Adults who have a strong expectation that spades are black might take longer to identify it correctly or even misperceive the color initially.
Whereas kids without that strong learned expectation might see it accurately more quickly.
Right. Their perceptual set isn't as rigidly formed yet for that specific stimulus.
The sources also connect perceptual set to social perception, mentioning studies like Keith Payne's and real-world incidents. They neutrally present the research finding that, under time pressure, exposure to faces of one ethnicity versus another could influence the speed or likelihood of misidentifying an ambiguous object, like a tool, as a weapon.
Yeah, showing how these basic perceptual mechanisms can potentially interact with learned associations or biases in complex social situations. It's presented as content from the source material.
Okay, one more way we manage sensory input, sensory adaptation, getting used to things.
Exactly. It's a decrease in sensitivity to a stimulus that remains constant over time. Your sensory receptors essentially become less responsive to unchanging input.
Like walking into a room with a strong smell overpowering at first, but after a few minutes you barely notice it.
Or the feeling of your clothes on your skin, the hum of the refrigerator, adjusting to the water temperature in a pool. Your senses adapt to the average level of stimulation.
It takes time, though. Like driving out of a dark movie theater into bright sunlight, you're temporarily blinded while your eyes adapt.
Right. Adaptation is useful because it allows our senses to remain sensitive to changes in the environment, which are often more important for survival. But the trade-off is that reduced sensitivity to the constant stuff and the time it takes to readjust when conditions change drastically.
Okay, let's get into the specific senses, starting with the big one, vision.
Right. The stimulus for vision is light, which is electromagnetic energy, and its properties determine what we see.
So wavelength determines the color, the hue. Visible light is just that tiny slice of the spectrum, 400 to 700 nanometers or so.
Yep. And the amplitude or height of the light wave determines its perceived brightness. Taller waves, brighter light.
And purity of the wavelengths relates to saturation, right? How rich or vivid the color is. A pure red is highly saturated. Pink is less saturated because it has white light mixed in.
Exactly. And the eye is this incredible biological camera designed to capture and focus this light energy. Figure 3.8 shows the main parts.
You've got the white outer layer, the sclera, giving it shape. Then the colored part, the iris, which controls the size of the pupil.
The pupil is the opening, like the aperture on a camera, letting more or less light in depending on the conditions.
And the focusing is done mainly by the cornea, that clear front bulge that does most of the bending, and then fine-tuned by the lens behind it.
Right, the lens is flexible, changing shape to focus on objects at different distances, though it gets less flexible as we age, hence needing reading glasses.
And all that focused light lands on the retina at the back of the eye.
Yeah.
That's the light-sensitive layer, the film in the camera analogy.
Yeah, the retina is where the crucial transduction happens. It's packed with photoreceptor cells, about 126 million of them. And there are two main types.
Rods and cones?
Rods are way more numerous, around 120 million. They're incredibly sensitive to light, so they're essential for vision in dim conditions, like at night, but they don't detect color.
And they're found all over the retina except the very center.
Right, except in the fovea. So that trick for seeing a faint star, look slightly beside it, that puts the light onto the rods in your peripheral retina, which are sensitive enough to pick it up.
And cones. There are fewer of those.
Much fewer, only about 6 million. They're responsible for color vision and for seeing fine detail, but they need much more light to work well, so they function best in daylight or bright indoor light.
And they're concentrated in the fovea, that little pit in the center of the retina.
Exactly. The fovea is almost pure cones. That's why your vision is sharpest when you look directly at something you're using your fovea. Try reading this text using only your peripheral vision. It's really difficult because there are far fewer cones out there.
Okay, so light hits the rods and cones. Then what happens? Figure 3.10 shows the pathway.
The rods and cones convert light energy into electrical signals. These signals pass through intermediate cells called bipolar cells and then onto ganglion cells.
And the axons of these ganglion cells bundle together.
To form the optic nerve, which exits the back of the eye, carrying all that visual information towards the brain.
And the spot where the optic nerve leaves, That's the blind spot.
Correct. No rods or cones there, so you're technically blind in that small area of your visual field. Figure 3.11 has that exercise to demonstrate it.
But we don't notice a hole in our vision.
Exactly. Because your brain, using top-down processing, brilliantly fills in the gap. It uses information from the surrounding area and basically makes its best guess about what should be there. Another example of perception being an active construction.
So the optic nerve carries the signals. Where to next?
The optic nerves from both eyes meet at the optic chiasm. It's a crossover point. Figure 3.33 shows it.
What crosses over?
About half the fibers from each eye. Specifically, the fibers from the nasal side of each retina, closer to your nose, cross over. The fibers from the temporal side, closer to your temples, stay on the same side.
Okay. So what does that mean for what the brain sees?
It means that everything you see in your left visual field, the entire left half of your view, gets processed by the right hemisphere of your brain. And everything in your right visual field gets processed by the left hemisphere.
Regardless of which I saw it initially.
Right. The information is sorted by visual field, sending it to the opposite brain hemisphere, ultimately landing in the visual cortex in the occipital lobes at the back of the brain.
And in the visual cortex, the real detail processing happens, like identifying specific features.
Yeah, through feature detectors. These are specialized neurons discovered by Hubel and Wiesel in their Nobel Prize-winning work that respond selectively to very specific features in the visual input lines at a certain angle, edges, corners, movement in a particular direction.
It suggests the brain breaks down the visual scene into these basic components first. And their work also hinted at critical periods for development.
Yes, that early visual experience seems crucial for these feature detectors and the visual system wiring to develop properly. The case of Michael May, who gained sight as an adult, is mentioned his brain had to essentially learn to interpret the visual input, initially seeing patterns rather than distinct objects.
And the brain processes all these different features, shape, color, motion, depth, all at once.
Yes, through parallel processing, it doesn't analyze shape, then color, then motion. It farms out these tasks to different neural pathways that work simultaneously. It's incredibly efficient.
But then how does it put it all back together? Seeing a single integrated object like a red ball rolling.
That's the binding problem. It's a major puzzle in neuroscience. How do all those separate features, processed in different brain areas, get bound together into a unified perception? The leading idea involves the synchronized firing of neurons across those different areas.
Okay, let's talk color vision. It makes the world so much more vibrant.
And it's evolutionarily useful, think, spotting ripe fruit against green leaves. But remember, color isn't in the light itself. It's how our brain interprets the signals from our photoreceptors in response to different wavelengths.
There are two main theories, right? Trichromatic theory first.
Right. This theory proposes we have 3 types of cones, each maximally sensitive to different wavelengths, roughly corresponding to short, blue, medium green, and long red wavelengths. All the colors we perceive are created by the combined pattern of activity across these 3 cone types.
Like mixing primary colors of light. And this explains color blindness.
It does, yes. Most common forms of color blindness involve a malfunction in one or more of these cone types, typically the red or green cones.
Okay, then there's the opponent process theory.
This theory suggests that color vision operates through pairs of opposing color channels in the visual system, specifically red-green, blue-yellow, and also black-white for brightness. Certain cells might be excited by red and inhibited by green, or excited by blue and inhibited by yellow.
And this explains things like after images. Stare at a green patch, then look at white, and you see a red after image.
Exactly. The green channel gets fatigued, so when you look away, the opposing red channel rebounds. It also explains why we can't perceive impossible colors like reddish-green or bluish-yellow. The channels oppose each other.
So, which theory is right?
The cool thing is both are right. They operate at different stages. The cones themselves respond according to trichromatic principles, but the signals from the cones are then processed by later neurons, like ganglion cells and cells in the thalamus and cortex, in an opponent process manner. Figure 3.16 shows how the 3 cone signals get recoded into opponent channels. The visual system cleverly uses both strategies.
Wow, okay. Beyond color, how do we perceive shape, depth, motion, and constancy?
Well, shape perception often starts with detecting contour, which is basically an edge or boundary where brightness changes abruptly. A fundamental organizing principle is the figure-ground relationship.
Like the classic goblet faces illusion in figure 3.17, we see either the white goblet as the figure against a black ground or 2 black faces as the figure against a white ground.
Exactly. Our brain automatically tries to separate the main object figure from the background, ground. This is related to the Gestalt principles of perception.
Gestalt meaning whole or form.
Right. The idea that we perceive objects as organized patterns or wholes, not just collections of separate sensations. The whole is different from the sum of its parts. Principles like closure, filling in gaps, proximity, grouping nearby items, and similarity, grouping similar items, shown in Figure 3.18, help us organize the visual world automatically. Movies rely on this perceiving motion from still frames shown rapidly.
What about depth perception? Seeing in 3D from the 2D images on our retinas.
We use a variety of cues. Binocular cues require both eyes. The main one is binocular disparity.
The difference between the images each eye sees.
Exactly. Because your eyes are set slightly apart, they get slightly different views of the world. Your brain compares these two images, and the degree of difference or disparity tells it how far away objects are. More disparity means closer object. The hand demonstration illustrates this well.
And convergence.
That's the extent to which your eyes turn inward when focusing on a close object. Your brain monitors the muscle tension involved and uses that as another cue for distance.
But we can still perceive depth with one eye closed, right? Using monocular cues.
Oh, absolutely. Monocular cues are very powerful. The sources list several. Familiar size. If you know how big an object usually is, you can judge its distance. Height in the visual field. Objects higher up often look farther away. Linear perspective. Parallel lines appear to converge in the distance. Relative size. Closer objects look bigger. Overlap. An object blocking another is closer. Shading. Patterns of light and shadow give clues to form. And texture gradient. Textures look finer and denser as they get farther away. Figures 3.19 and 3.2z show some of these.
The story of Susan Berry, who gained stereo vision later in life, really highlights the difference binocular cues make. She described the world suddenly looking richer, more inflated.
Yeah, it emphasizes that while monocular cues give us a good sense of depth, true stereoscopic vision adds another layer of realism and spatial understanding.
And motion perception. How do we see things move?
It's critical for survival. Our brains have specialized neurons tuned to detect movement. We also use feedback from our own eye and head movements and compare the changing position of objects relative to stationary backgrounds. Sometimes we perceive motion when there isn't any, apparent movement in movies or feeling like you're moving in an IMAX film.
Finally, perceptual constancy. This seems crucial.
It is. It's our ability to perceive objects as stable and unchanging in size, shapes, brightness, color, even though the sensory information we receive from them changes constantly as our viewing angle, distance, or lighting conditions change.
So a door still looks like a rectangle even when it's open, and the image on my retina is a trapezoid. That's shaped constancy.
Exactly. And a car driving away looks like it's staying the same size, even though its retinal image is shrinking size constancy. An apple looks red under fluorescent light or sunlight color constancy. Your brain is actively interpreting the raw sensory data in context to maintain a stable perception of the world.
But these constancies can be tricked by illusions, right? Like in figure 3.23.
Yes, illusions often exploit these constancy mechanisms or depth cues, presenting them in unusual contexts where they lead our perception astray. They show how adaptive these processes usually are, but also that perception is an interpretation, not a perfect copy of reality.
Okay, let's switch senses. Time for the auditory system and the world of sound.
Right. Sound travels as sound waves, which are vibrations moving through a medium, usually air for us,
And the properties of those waves determine what we hear. Wavelength determines frequency.
which we perceive as pitch. Higher frequency, higher pitch. Lower frequency, lower pitch. Figure 3.24 shows this.
And amplitude of the wave.
That determines loudness, measured in decibels or dB. Higher amplitude, louder sound.
Most sounds aren't pure tones, though. They're complex mixtures of frequencies.
Right. And that complexity gives a sound its timbre, or tone quality. It's why a trumpet sounds different from a violin playing the exact same note at the same loudness. It's the unique blend of frequencies.
The safe sound info in the sources is a good practical note, basically. Watch the volume and how long you listen, especially with headphones or in noisy places where you tend to crank it up.
Good advice. Now, the ear itself is structured to capture and process these sound waves. Figure 3.25 shows the three main parts.
The outer ear, that's the visible part, the pinna, which funnels sound into the auditory canal.
Yep. Then comes the middle ear. Sound waves hit the eardrum, making it vibrate.
And those vibrations are passed along those three tiny bones, the smallest in the body.
The hammer, anvil, and stirrup. They act like a lever system, amplifying the vibrations and transmitting them to the oval window, which is the entrance to the inner ear.
Why amplify?
Because the inner ear is filled with fluid, and it takes more force to move fluid than air, so the middle ear bones boost the pressure. They also have tiny muscles that can tighten up to slightly reduce the transmission of very loud sounds, offering some protection.
Okay, then the inner ear. This is where the conversion to neural signals happens,
Yes, vibrations enter through the oval window into the cochlea.
which looks like a snail shell.
Exactly, it's a coiled, fluid-filled tube. Running through the center of the cochlea is the basilar membrane. When sound vibrations enter the cochlea, they cause waves in the fluid, which in turn make the basilar membrane move.
And lining the basilar membrane are the crucial cells.
The hair cells. These are the actual sensory receptors for hearing. They have tiny hair-like projections called cilia. When the basilar membrane vibrates, these cilia bend against another membrane called the tectorial membrane.
And that bending triggers the neural impulse.
That's the transduction step for hearing. That bending generates the electrical signal that gets sent to the brain via the auditory nerve. But these hair cells are incredibly delicate.
And damage from loud noise is often permanent because they don't regenerate.
Correct. That's why protecting your hearing is so important. Cochlear implants work by bypassing damaged hair cells and directly stimulating the auditory nerve fibers.
How does the cochlea figure out the pitch, the frequency of the sound? There are theories about this too.
Yes. Place theory, proposed by Georg von Békessy, suggests that different frequencies cause the peak vibration at different places along the basilar membrane. High frequencies vibrate the base of the membrane most, while low frequencies vibrate the tip most. The brain knows the pitch based on which place is most active. This explains high-frequency hearing well.
But what about low frequencies?
That's where frequency theory comes in. It proposes that the perception of pitch corresponds to the rate or frequency at which the entire basilar membrane vibrates, causing the auditory nerve to fire at the same rate. So a 100 Hz sound makes the nerve fire 100 times per second. This works well for low frequencies.
But individual neurons can't fire fast enough for really high frequencies.
Right, so the volley principle modifies frequency theory. It suggests that for frequencies above what a single neuron can handle, groups of neurons work together, firing in alternating bursts, or volleys. Their combined firing rate matches the sound frequency, so it seems the brain uses both place coding for highs and frequency volley coding for lows and mid-range.
And the signals travel along the auditory nerve to the brain.
Yep. The pathway is complex, involving several stalks in the brainstem and thalamus before reaching the auditory cortex and the temporal lobes. And like vision, there's crossover, with most information from one ear going primarily to the opposite brain hemisphere.
One last thing on hearing. Localizing sound. How do we know where a sound is coming from?
Having two ears is crucial. Your brain compares the information arriving at both ears.
What kind of information?
Two main things: timing differences and intensity differences. A sound coming from your right will reach your right ear slightly sooner than your left ear. It will also be slightly louder in your right ear, especially for higher frequencies, because your head creates a sound shadow, blocking some of the sound waves from reaching the far ear. Figure 3.27 shows this.
And the brain calculates these tiny differences to figure out the direction.
Exactly. It's very precise. Although it's harder to localize sounds that are directly in front, behind, or straight above you, because the timing and intensity cues are ambiguous then.
Some animals are much better at this, aren't they? Like bats.
Oh, bats are masters. They use echolocation, emitting high-frequency sounds and interpreting the returning echoes to navigate and hunt in complete darkness. It's an incredible sensory adaptation.
Okay, let's broaden out to the other senses. Touch, taste, smell, body awareness. Starting with the skin senses.
Also called the cutaneous senses. Our skin is our largest sensory organ, packed with receptors for touch, pressure, temperature, and pain.
Touch detects mechanical energy, pressure against the skin, finding keys in your pocket, feeling the texture of fabric.
Right. Signals travel from receptors in the skin up through the spinal cord, cross over in the brainstem, go through the thalamus, and end up in the somatosensory cortex in the parietal lobe. And sensitivity varies hugely. Your fingertips are way more sensitive than, say, your back.
And touch is really important for development, especially in infants.
Absolutely. Exploration, comfort, attachment. And interestingly, the sources note that very intense stimulation of any sense can sometimes be felt as pressure or even pain. Like a painfully loud noise feels like pressure.
What about temperature?
We have specialized thermoreceptors under the skin, separate ones for detecting warmth and detecting cold relative to your skin's current temperature. They help regulate body temperature. And figure 3.28 shows that cool experiment. Stimulating nearby cold and warm spots simultaneously can create a paradoxical sensation of intense heat.
And pain. Not pleasant, but vital.
Absolutely essential. Pain is the body's alarm system, signaling damage or potential damage. Receptors for pain, called nociceptors, are found throughout the body and respond to various stimuli: intense pressure, extreme heat, or cold, damaging chemicals released by injured tissues.
And there are different pain pathways.
Yes. A fast pathway uses thicker myelinated nerves to send signals quickly to the thalamus and cortex. This carries sharp, localized, immediate pain, the initial ouch warning. A slow pathway uses thinner, unmyelinated nerves and travels through parts of the limbic system. This carries the duller, throbbing, longer-lasting, more unpleasant ache that keeps reminding you you're injured.
And the experience of pain isn't just about the receptors firing.
Not at all. The brain constructs the experience of pain. It's heavily influenced by things like attention, expectation, emotional state, past experiences, and even cultural factors. The sources mention rituals where people seem to endure extreme stimulation without recording pain. And the brain has its own pain relief system involving endorphins, natural opiates, especially linked to the slow pathway.
Which is why psychological factors and treatments are so important for chronic pain. And simple things like distraction or counterstimulation rubbing a sore spot can actually reduce perceived pain.
Exactly. Pain perception is incredibly complex and subjective.
Okay, on to the chemical senses. Taste and smell, they work together closely.
Very closely. Much of what we perceive as flavor actually comes from smell. That's why food tastes so bland when you have a cold and your nose is blocked.
Taste or gustation happens on the tongue.
Yeah.
The bumps, papillae contain the taste buds.
Right. We have about 10,000 taste buds, and they contain the receptor cells. Interestingly, these cells get replaced fairly often, about every 2 weeks. Signals go from the taste buds to the brain.
We usually talk about the basic tastes. Sweet, sour, salty, bitter. Figure 3.29 shows rough areas of sensitivity.
Yes, those are the traditional 4. But it's more complex. Many taste cells respond to multiple types of chemicals, and the brain integrates the overall pattern of signals. Plus, there's strong evidence for a 5th basic taste, umami.
The savory taste.
Exactly. Detected by receptors sensitive to glutamate, an amino acid found in protein-rich foods like meat and cheese, and also added to things like chips as MSG, gives that satisfying, savory flavor. And culture hugely shapes our taste preferences.
What about smell or olfaction?
The receptors for smell are located high up in the nasal cavity in a patch of tissue called the olfactory epithelium, figure 3.30. When we sniff, we draw air and odor molecules up to these receptors.
And these olfactory neurons are unusual because they can regenerate.
Yes, they're one of the few types of neurons that regularly replace themselves throughout life, which is good since they're fairly exposed.
And the pathway to the brain is different too.
Very different. Unlike other senses, olfactory information doesn't primarily go through the thalamus. Instead, it has a fairly direct route to olfactory processing areas in the temporal lobe, and importantly, very strong connections to the limbic system brain areas, heavily involved in emotion and memory, like the amygdala and hippocampus.
Ah, so that's why smells can trigger such strong, sudden memories and emotions. That direct link.
That's the leading explanation, yes. It's like a sensory superhighway to the emotional and memory centers of the brain. The sources also briefly mention research exploring smell's potential role in attraction, possibly linked to detecting immune system compatibility through body odor.
Okay, last group. The senses that tell us about our body's position and movement. The kinesthetic and vestibular senses.
Right. These work together to give us proprioceptive feedback, our sense of where our limbs and body are in space and how they're moving.
The kinesthetic senses have receptors where?
In our muscles, tendons, and joints. They constantly feed information back to the brain about limb position and movement. You usually don't notice it unless it's gone, like when your leg falls asleep. But it's crucial for any coordinated movement from walking to playing piano.
And the vestibular sense is about balance.
Yes, balance and sensing head movement and orientation relative to gravity. The main receptors are in the semicircular canals in the inner ear near the cochlea. Figure 3.31 shows them.
3 fluid-filled canals.
Right, oriented in 3 different planes. As your head moves, the fluid inside these canals lags slightly, bending hair cells located within them. This signals the direction and speed of head rotation. Other structures, called vestibular sacs, detect linear acceleration and gravity.
And this information helps us keep our balance and coordinate eye movements with head movements. The pathway travels along part of the auditory nerve.
And that spinning experiment in figure 3.31 makes sense. Now spin around, then stop suddenly. The fluid in the canals keeps swirling for a moment, telling your brain you're still spinning, which conflicts with what your eyes and kinesthetic sense are saying, causing dizziness.
And vision plays a big role here too, right? Like feeling motion in an IMAX movie.
Definitely. Strong visual cues of motion can override or influence your vestibular sense, creating a powerful illusion of movement, or sometimes motion sickness if there's a conflict.
Wow. Okay, so looking back over everything we've discussed from these sources, sensation is really about the raw data collection, the initial detection of energy.
Yeah, the input stage, detecting light, sound, pressure, chemicals.
But perception is where the brain steps in as this incredibly active interpreter. Organizing, making sense, using context, predicting.
Absolutely. It's constructing your reality based on that sensory input plus all your prior knowledge and expectations. It's not just a passive window onto the world, it's more like an active artist painting a picture based on the light coming through the window, filling in gaps like the blind spot, maintaining constancy despite changing input.
So let's circle back to those first questions. How do these incredibly complex layered processes from the firing of a single receptor cell to these widespread brain interpretations actually create your experience of your favorite song or the taste of that amazing meal?
And maybe even more profoundly, how does this continuous, deeply personal process of sensing and perceiving the world literally shape who you are, your sense of self interacting with reality?
Right. And building on everything we've talked about, perception is interpretation, the brain filling in gaps, perceptual sets filtering reality, constancy creating stability.
Yeah.
Here's something to really chew on.
Given that your brain isn't just passively receiving objective information, but is actively selecting, organizing, interpreting, and even guessing based on expectations, how much of what you experience, with complete confidence, as external objective reality?
There's actually unique subjective construction happening entirely inside your own head, moment by moment.
Something to think about next time you, well, sense or perceive anything.